Hello everyone. I hope you guys are doing well. On your demand, I have brought for you another data science project which involves the use of graph neural networks. So the problem that we are going to be talking about in today's machine learning project is to predict lipophilicity using graph neural networks. The dataset that we will be using is curated from Campbell database, which contains experimental results on octanol water distribution coefficient or log D at pH 7.4. So basically, this is our target log D, which is based on experimental values of the octanol water distribution coefficient. And lipophilicity is crucial in drug discovery as it significantly influences a drug's absorption, distribution, metabolism, and excretion properties. Optimal lipophilicity enhances a drug's ability to cross cell membranes, affecting its bioavailability and therapeutic efficacy. However, excess lipophilicity can lead to poor solubility and undesirable interactions with biological molecules, potentially causing toxicity. So lipophilicity is important, and that's what we are going to be trying to predict in today's project. For this series of lectures, I will be talking about first data analysis using pandas, matplotlib, seaborn, and rdkit. Then we will move on to generating graphs from smiles using Pytorch geometric. Then we will do some graph analysis using networkx, train test split using Pytorch. Then we'll define training and evaluation functions, which are one of the most important aspects of graph neural networks. And then we will move on to model training and evaluation, again using Pytorch geometric. Then we will also see how to do hyperparameter tuning using Optuna. And then in the end, we will do some post-hoc analysis, and then we will compile all our results in the form of a manuscript for us to communicate somewhere. Just a little bit about graphs. This is acetic acid molecule, and this is the graphical representation of this molecule, where these atoms, for example, this carbon, oxygen here, are represented as nodes, and the bonds between them are represented as edges. Now, of course, computer is not really gonna understand these things. We need to input numbers for us to train our models, right? So if we want to visualize how the graph-based descriptors would look like, then we can do a little comparison with the traditional methods of representing a molecule. For example, fingerprints. This is MAC's keys, and it has various keys, and its values are in the form of zeros and ones. Similarly, if we have rdkit descriptors, these are all global descriptors. Basically, this is the molecular weight, and it represents the molecular weight of the whole molecule here. And O count basically represents the number of oxygens, which are 2. SPS is another descriptor. Number of valence electron is 24. TPS is another descriptor here with a value of 37.3. So all these traditional descriptor represents the global attributes of these molecules, which we can train using different models like linear regression or random forest or even neural networks. Now, when it comes to graphs, we need a lot of information. For example, we need to know the connectivity between all these atoms, and the connections are represented by adjacency matrix. So on the left hand side of the table, and on the top we have the labeling of these atoms. For example, this carbon is carbon 0, this carbon is carbon 1, and we want to see the relationship between them, whether 0 and 3 or 1 or 3 are connected and not, and that can be done with the help of this adjacency matrix. Basically, if we look at the first row here, there is no connection between 0 and 0, but there is a connection between 0 and 1. There is a single bond. So we have a 1 here, and rest all are 0 values. Similarly, between 1 and 0, we have a bond. 1 and 3, we have a bond, and 1 and 2. So basically there are 3 bonds, so 3 connections are there, so 3 ones, and the connection with itself is basically 0. Similarly, for second label here, which is oxygen, we just have a connection with this carbon here. So carbon atom 1, so we have 1 here, rest all values are 0. Similarly for this oxygen here, which is labeled as 3, we just have a connection with carbon atom 1, and all other values are 0. So adjacency matrix basically tells us the connections. And then we have node features or node attributes or atom properties. For example, this carbon, if we think about the properties associated with the atoms, there could be many properties. For example, atomic weight, atomic formal charge, and stuff like that. Now the first property that I'm showcasing here is that of atom type. Now this could be here, this is carbon, but in general we can have carbon, hydrogen, oxygen, nitrogen, sulfur, and that's what we have on here. Basically we are gonna be one hot encode all these atoms in here based on the atom type. If the carbon is here at position 0, we will have 1. So we have 1 here because at labeling 0 and 1 we have carbons, and all rest of these are 0. Similarly at position 2 we have oxygen, so during one hot encoding we have 1 here and rest all are 0. Similarly this is also oxygen, so this is 1 here. Another property which could be attributed to the atoms is formal charge. Now this is a neutral molecule, so formal charge is 0, so all these values are 1 here and rest all are 0. Similarly atomic weight, like I told you before, this carbon has an atomic weight of 12, this is also 12, this is 16, so all these numbers are here. Now you can also scale these values if you want. So similarly there are many other properties which you can include in the graphical representation, and this is basically called node feature matrix. Now apart from nodes, we also have edge attributes or edge features or properties of the bonds. And again, if we think about the bond properties, it could be a single bond, double bond, triple bond, and that's what we have over here. We have a bond type, and we are gonna be encoding these one hot encode these based on the bond type, whether the bond is single bond, double bond, or triple bond. If the bond is single, we will have a value of 1, and that's what we have between 0 and 1 and 1 and 2. So 0 and 1 is a single bond. Similarly 1 and 2 is a single bond, so we have 1 here, and all these values are 0. Whereas between 1 and 3 we have a double bond, so this value is 1, whereas all these values are 0. So this is basically a representation or graphical representation of a single molecule. And let's say if we have 100 molecules of 1000 molecules, we have graphical representation of all of these atoms and bonds in a similar manner, in the form of adjacency matrix, node feature matrix, edge feature matrix. And we are gonna be passing all these information in the convolution layers followed by linear layers to train our model and finally get our output in the form of our target, whichever target we are interested in. So that's what we are going to be talking about in the series of lectures. So let's just go back to the Google Colab notebook and let's just start coding. So we are going to start off by importing installing two important libraries, which is RDKit and Pytorch geometric. So this is the syntax for that. So let's go ahead and install these, and along with that I'm also going to be importing some traditional libraries for data manipulation and visualization, such as NumPy, Pandas, Matplotlib, Seaborn, NetworkX, and some important modules from RDKit and from the Torch and Torch geometric. So let's go ahead and import these as well. Now since we are going to be plotting a lot, so I am just using ggplot as our style. You can print out these different styles here using this command, and it will print out all the styles available, and you can choose your style or the style of your choice. I'm just using ggplot, so let's run this. So for reproducibility, I am setting up a random seed of 60. We can do 0 or 50, it just does not matter. But for the reproducibility, we need to have a random seed, and we have a random dot seed, os dot environ, Python seed, NumPy random seed, Torch seed, and all these things will run the same if you use the same value of 50 in your collab notebook as well, and it will give us the kind of same numbers. So this is a function, and we are going to be calling this function here. Now the first step is loading our data set. I have the lipophilicity.csv file in the temporary folder, and that's what I'm gonna be using, and we are gonna be loading using pandas, and I'm storing it in a variable called df. So let's just have a look at the head or the first 5 data points. So we have the Campbell IDs, experimental log D values, which I already talked about, and the smile strings. So that's all we have, and we are gonna be using these smile strings for generating graphs. And then once we generate the graphs, we are going to be training our model using graph neural networks. But first, let's just analyze our data set. Now I'm calling a describe method. So basically we have a count of 4200. So we have 4200 molecules. This is the mean and standard deviation. The range is between -1.5 to 4.5. So it's pretty good. And this is the medium 2.36, and let's just see if there are any duplicates. For that, I'm just using an unique method, and it should give us 4200 if there are no unique values. So the number is 4200, so there are no duplicates. Now the first thing is distribution plot, whether it is normally distributed or not. So that's what I'm gonna be visualizing with the help of Seaborn. So first I am using a figure size of 6 by 6, and then I'm using SNS or Seaborn and histplot method. The data is df over here, which we have uploaded, and the x-axis is our experimental log D values. So yeah, that's pretty much it. So let's run this. So this is the distribution plot. So it is pretty good. We have a kind of standard distribution. It's not perfectly normal, but still I think we have a pretty good data set in terms of our target values. And now let's just move on. And I also want to see the distribution of molecular weights. I want to see the lowest and the highest molecular weight, and I want to see the distribution, basically how many molecules have a molecular weight range of what. So for that we need to convert these smile strings into molecules, and I have talked a lot about this in my previous videos, especially the videos on RDKit. So you can use this pandas tool method in RDKit and add molecular column to frame, which would take in the smile strings for the data set, and it will generate another column called mole, which would have the molecular objects, right? So let's just run this and have a look at our new data set here. So initially we had 3 columns. Now we have another column called mole, which have molecular objects, and we are going to be using this molecular objects to generate the another column of molecular weight. And for that I'm just using RDKit. I just highly recommend you, if you haven't seen the RDKit videos, I have made 2 videos on that. I think it would be highly beneficial for you if you go and watch those videos. I have given brief introduction and description about how you can generate different descriptors and how you can analyze your data using RDKit. So what I'm doing here is that I'm generating an empty list called molecular weight, and I'm looping through each molecular object, and with the help of descriptors dot molecular weight, I am generating the molecular weight of these molecules, and I am appending the molecular weight to this empty list here, and in the end we'll have a list of 4200 molecules containing their molecular weights, which we can convert or append it to our data frame here. So let's run this and have a look at our data set again. So now you can see here we have the molecular weight of each compound here. Now similar to above, all I'm doing is just replacing this. Initially we had x as our experimental log d values, so I'm just replacing log d values with molecular weight. Rest of the code is same. So let's run this again. So this is the distribution plot of molecular weight of these compounds, and as you can see, until now this is pretty good, but we have few molecules, just few molecules with these molecular weights, like around 850, this is around 970, something like that. So these are very few molecules, and let's just visualize these molecules, right? So we want to know what kind of molecules we have which have just such a high molecular weight. So basically I'm just slicing a data set based on molecular weight. So df molecular weight greater than 700, it will throw me a boolean values, and based on that we will slice our data set. And these are the compounds with high molecular weight. So this compound at index position 364 has 700 molecular weight, and so on. So now let's just visualize these molecules. Now this data set is stored in a variable called high_MWT, and what I'm using next is draw.mol to grid image, which would print out the molecular structures in the form of a grid. And again, why I'm using this data set here, which we have just saved, and molecular object column, number of molecules that we want to print in one row is 4. This is the size, and I also want to see the molecular weight as well. So the legend is the molecular weight. So let's run this. So these are the molecules with molecular weight greater than 700, and now what we can do is, since this is not really a normal distribution, there are just very few molecules with high molecular weight. So what we can do is we can also get rid of these molecules, because if we look at the shape, so we have 8 molecules with molecular weight greater than 700, and out of 4200, if we just get rid of 8 molecules, I don't think that's really gonna impact much. So what I'm gonna do is I'm gonna remove these molecules from our data set. So these are the smile strings that we wanna get rid of from our initial data set. So that's what I'm doing here. I'm just getting rid of these molecules from our original data set, and I'm storing the new data set in a variable called df_final. So let's run this, and this would be our final data set. And if you want to look at the shape, so this is 4192 molecules. So we have got rid of the 8 molecules here, and we can again plot the distribution plot for the molecular weight and see how it looks now. So it looks pretty good, and it is quite normal distribution. So yeah, now this is the final data set that we are gonna be working on. Again, I want to reset the index here, and now you can see in here the count is 4192, So the maximum molecular weight is 697.77 and minimum molecular weight is 113. So this was all about distribution plot, and let's just print out our final data set here again. Now what we want is, I also want to see what types of atoms are there. Basically the frequency of occurrence of elements in the data set. So what I'm doing here is that I'm creating a set of unique atoms. So this is an empty set, and what I'm doing is iterating through each molecular objects here. So this is the molecular object, and using RDKit I am iterating through each atom in the molecule, and I'm getting the symbol of this molecule, and the symbol would be added to this set, and there would be no repetition because this is a set. And we'll just even though there would be elements which would occur multiple times, but the set would just give us only unique atoms. So let's run this and see how many unique atoms are there in our data set. So we have a total of around 1 2 3 4 5 6 7 8 9 10 11 and 12 elements are there in our data set. Of course, if you get rid of carbon, we have 11 hetero atoms here. And I want to see the frequency of each of these. I want to see how many times nitrogen is there in the data set. For example, if the first molecule has occurrence of nitrogen 5 times, the second molecule has 3 times, and the third molecule has 2 times. So 5 plus 3 plus 2 and so on. And so basically the frequency of occurrence of heteroatoms in the whole data set. For that, again, I am just creating an empty list, and using RDKit we will iterate through each molecule and through each atom, and we will just store the symbol of that atom in the atom list. And in the end we will use the counter function to generate a dictionary. So basically this is the dictionary here, and carbon is occurring 83,000 times. Again, these are hydrocarbons, right? So that's not really relevant to us. So I have just copy pasted all other elements here along with the number of times it has occurred. So nitrogen has occurred 14,000 times, and it kind of makes sense because the drugs they need to be kind of soluble, so we need to have some sort of heteroatom. So nitrogen has occurred this many times, and then I think it's oxygen. So how about we just plot this in the form of a bar plot for better visualization. For that, first I'm converting it into a data frame. So df underscore freq okay, so this is the data frame, and we can plot a bar plot using SNS. So first I am here creating a figure object, and then using SNS dot bar plot, the axis the atom symbols and y is the count. So let's just run this. So this is kind of the bar plot, and as you can see the nitrogen has occurred more than 14,000 times in a data set containing 4,200 molecules, and oxygen is at the second spot with little more than 10,000 times occurrence. Fluorine is little less than 2,000 times, sulfur and chlorine again little more than 1,500, and all other atoms are quite less, like bromine, iodine, boron, phosphorus, silicon and selenium. And if we go above, you can see silicon and selenium, they have occurred just only once, and so on. So mainly nitrogen, oxygen, fluorine, sulfur and chlorine, that kind of make up most of our data set. Now this is the frequency of occurrence of these atoms in the whole data set, right? Now if we want to see the number of molecules which contain these atoms, for that this could be a little complicated syntax, but what I'm just doing here is this is the reference. These are the unique atoms that we have in our data set, right? And what I'm just doing here is, with the help of RDKit, I am just checking if this element is present in a molecule, and if it is present, I am just increasing the count, and that's pretty much it. And if we run this, and if we look at our dictionary 2, basically this is kind of the number of molecules. Of course there would be overlapping as well, so let's just first convert it into a data frame and then plot a bar plot, and then we'll see or analyze our results here. So you can see here, so now these are the molecules. So basically in more than 4,000 molecules we have the occurrence of nitrogen and oxygen just at the second spot here. Now of course there could be overlapping as well. For example, the nitrogen containing molecules can have oxygen atom as well and sulfur as well, so there's definitely gonna be overlapping, but these are kind of the molecules which do have this nitrogen and oxygen and sulfur, so on. And yeah, I think that was pretty much it in terms of data analysis. And in the next lecture we will be generating and analyzing graphs using Pytorch geometric, and then we will analyze using network X. So that is it for now. Please let me know in the comment section if you have any questions. Thank you for watching.